Hello friends, good afternoon and welcome to EduSite Live Lectures. Dear friends, today in food microbiology, we will be talking about food intoxication. To discuss this topic, we have with us our subject expert, Dr. Pavas Goswami. Dr. Goswami is Assistant Professor in Department of Microbiology in Bhaskaracharya College of Applied Sciences, University of Delhi. Without further ado, I would like to welcome Sir to our studios and request him to start the lecture. Welcome, sir.
Thank you. Good afternoon, dear viewers. Today I will be talking about food intoxication, and in this I will be talking about two bacteria, Staphylococcus aureus and Clostridium botulinum. So, in my lecture today I will be discussing some definitions, food poisoning, food intoxication, gastroenteritis and Staphylococcus aureus, its important characteristics, antigenic structure, enzymes and toxins, habitat and distribution, enterotoxins produced by this bacteria, symptoms of staphylococcal food poisoning, treatment, foods involved, ecology and its prevention. Term food poisoning as applied to diseases caused by microorganisms is used very loosely to include the illnesses caused by ingestion of toxins elaborated by microorganisms and those resulting from infection of host through intestinal tract. This will be clear with these animations. These are schematic animations for the disease. So, disease can occur in many ways. First of all, a food pathogen may enter the body through food, and when it will enter, it can infect the cells, basically intestinal cells when we talk about gastroenteritis, and when it will infect these cells, this will multiply inside and cause disease. This is foodborne infection when live pathogen is present inside the body and is coming through food. Another scenario is when both the pathogen and toxin are present. These circles depict the toxins, and when these toxins are present along with this cell, they will cause damage to the cells, and this scenario is foodborne infection. In another scenario, food may contain preformed toxins, and these toxins will cause damage inside the body, and this scenario is foodborne intoxication. So, in food infection, live cells are present. In foodborne intoxication, toxin is present inside the food. Now, food intoxication is foodborne illness with toxin, and now we will take up staphylococcal food intoxication. Another term is gastroenteritis. This designation, gastroenteritis, obviates the need to indicate whether the illness is an intoxication or an infection. This will include both the foodborne infection and intoxication. Coming to Staphylococcus aureus, Staphylococcus aureus usually forms grey to deep golden yellow colonies. Gram positive cocci are present in bunches. As they are gram positive, they have high concentration of peptidoglycan in their cell walls. These bacteria are coagulase positive and also positive for the enzyme catalase. You can see in the figure, these are small cocci which are arranged like cluster of grapes. Coming to growth characteristics and resistance of bacteria, staphylococci slowly ferment many carbohydrates producing lactic acid but not gas. Staphylococci are relatively resistant to drying, heat, and they can withstand 50 degrees centigrade for 30 minutes and 9% sodium chloride. These are facultative organisms. The media which is used for isolation of staphylococci is called mannitol salt agar, Its composition is proteose peptone, meat extract, sodium chloride, mannitol, phenol red, and pH is around 7.4. The important components of this medium are mannitol, which is fermented by Staphylococcus aureus, phenol red, which is a pH indicator and indicates that whether acid is being produced by mannitol or not. And another most important component of this media is sodium chloride. Because Staphylococcus can tolerate high concentrations of sodium chloride, so this has been used as a selective factor to choose or to isolate Staphylococcus from the food products, because other organisms which are not tolerating salt will be inhibited. So, by this mechanism Staphylococcus aureus can be selected from this. Here is a plate of mannitol salt agar. Now, if you observe, the original media is having pink color, which is present here in this streak. So, this bacteria is not utilizing mannitol and acid is not being produced, while all other bacteria which are these yellow streaks, they are producing acid by mannitol fermentation, resulting in the change in the color of the medium from pink to yellow. Another important growth characteristics and resistance is that of antibiotic resistance. Staphylococcus is known to produce enzyme beta lactamase, which inhibits antibiotics such as penicillin, penicillin G, ampicillin, ticarcillin, piperacillin and other drugs. Another important factor is resistant to nafcillin and methicillin oxacillin. This resistance is independent of beta lactamase production. Here the gene codes for penicillin binding proteins. These proteins are having low affinity for these drugs and thus these are not affected. So, antimicrobial resistance is being generated in two ways. One, Staphylococcus produce enzyme to change the structure, to destroy the structure of penicillin, beta lactam structure is destroyed by beta lactamase, and it also modify the target of penicillin, that is penicillin binding proteins. Bacteria is also resistant to tetracycline, erythromycin, aminoglycosides and other drugs, and this resistance is plasmid mediated, and this is a cause of worry because antibiotic resistance is increasing day by day in microbes, and vancomycin resistant strains of Staphylococcus aureus have been reported. And with increasing time, perhaps we will not have any drug of choice to cure these simple infections, leading to high mortality rates. Coming to the antigenic structure, this is having peptidoglycan, as the organism is gram positive. It is having high concentration of peptidoglycan in cell wall and lower concentration of lipids. This peptidoglycan elicits production of interleukin 1 in the body. It acts as chemoattractant for polymorphonuclear leukocytes and also activates complement. Another antigen is teichoic acid. This leads to adhesion of host sulfur. This helps bacteria in adhesion to host cell surfaces and protects from complement mediated opsonization. Another important antigen is protein A, which is a B cell mitogen. Staphylococcus aureus produce an array of enzymes and toxins. As discussed, catalase, coagulase are important enzymes produced by this. Hyaluronidase is produced, staphylokinase is produced, which help in spread of bacteria in the body. Then there are lipases, beta lactamase as I have discussed, hemolysins, leukocidin and enterotoxin. So, enterotoxin is the main toxin which is responsible for foodborne illness or foodborne intoxication caused by Staphylococcus aureus. Coming to habitat and distribution of Staphylococcus aureus, the largest numbers of Staphylococcus occur in anterior nares, axillae, inguinal area, then perineal area, and most domesticated animals are known to have Staphylococcus aureus. Milch animals suffering from Staphylococcus mastitis also harbor and shed Staphylococcus aureus. Coming to the temperature growth range, this organism is a mesophile. Some strains of Staphylococcus aureus can grow at temperature as low as 6.7 degrees centigrade. In general, growth occurs over a range of 7 to 47.8 degrees centigrade, but minimum and maximum temperatures of growth and toxin production assume optimal conditions relative to the other parameters. Various parameters affect the growth of bacteria. pH is there, then water activity is there, presence of compounds is there. So, if other parameters are optimum, then there can be variation in one parameter. So, bacteria will tolerate variation in one parameter much better if other parameters are optimal for growth. Then effect of salts and other chemicals on its growth. Staphylococcus aureus grow well in culture media without NaCl, but it can grow well in 7 to 10% concentrations of NaCl. As discussed in mannitol salt agar, we add NaCl at high concentration so that other strains can be inhibited while Staphylococcus aureus can grow. Some strains can grow in 20% NaCl. It has a high degree of tolerance to compounds such as tellurite, mercuric chloride, neomycin, polymyxin and sodium azide. Effect of pH. As discussed, pH is another important factor for growth of bacteria. Staphylococcus aureus can grow a pH range of 4 to 9.8, but optimum pH range is from 6 to 7. And again, if other parameters are optimal, then minimum growth pH and maximum growth pH can differ. Effect of water activity. Growth of Staphylococcus has been demonstrated as low as 0.83 water activity under otherwise ideal conditions, but 0.86 is the generally recognized minimum water activity for Staphylococcus aureus. Now, coming to the most important part, Staphylococcus aureus enterotoxins. These are potent gastrointestinal exotoxins. These are synthesized throughout the logarithmic phase of growth or during the transition from the exponential to the stationary phase. Let us discuss a bacterial growth curve. There are 4 phases of bacterial growth curve: lag phase, exponential phase, stationary and death phase. In lag phase, actual cell division do not take place, cell prepares to divide. In exponential phase, there is active cell division. In stationary phase, cell division is balanced with cell death, and in death phase again population starts declining. So, toxin production occurs in the exponential phase and the transition phase. Staphylococcus aureus enterotoxins are resistant to heat treatment, low pH, proteolytic enzymes, and it is active in nanograms to micrograms of quantity. Staphylococcus aureus enterotoxins belong to the broad family of pyrogenic toxin superantigens. Superantigens that induce vomiting after oral administration in a primate model are designated as Staphylococcus enterotoxins, and related toxins that lack emetic activity are designated as staphylococcal enterotoxin like S antigens. So, this is important. If a toxin is exhibiting emetic activity, then it is staphylococcal enterotoxin. If it is not exhibiting emetic activity, then this toxin is staphylococcal enterotoxin like S antigen. Several staphylococcal enterotoxins have been reported. These include classical staphylococcal enterotoxins such as SEA, SEB, staphylococcal enterotoxin C, Staphylococcus enterotoxin D and SEE. New types of staphylococcal enterotoxins include SEG, SEH, SEI, R, S and T. Staphylococcal enterotoxin like antigens include SELJ, K, L, M, N, O, P, Q and U. There is a very good review by Argudin et al. On food poisoning and Staphylococcus aureus enterotoxins. This has been published in Toxins, journal Toxins in 2010. So, you may refer it for more information. Staphylococcus aureus enterotoxins, among these enterotoxins, SEA and SED are more often associated with food poisoning outbreaks, and appreciable level of toxins are produced only after considerable growth of Staphylococcus. Enterotoxins are produced between 10 to 46 degrees centigrade, with the optimum temperature being 40 to 45 degrees centigrade. Enterotoxin production has been demonstrated in a good culture medium in 3 days at 18 degrees centigrade and in 12 hours at 37 degrees centigrade. So, you can observe at 18 degrees centigrade, when temperature maybe is not optimum for growth, it is taking 3 days for production of enterotoxins, while at 37 degrees centigrade, which is suitable for bacterial growth, and enterotoxin is being produced in 12 hours, and production of enterotoxin is more likely when competing microorganisms are absent or are few in numbers or inhibited for some reason. Presence of starch and protein in considerable amounts enhance toxic production. The enterotoxins are quite heat resistant. Crude toxin preparations have been found to be more resistant than purified toxins. Staphylococcus aureus cells are considerably more sensitive to heat than the enterotoxins. Pasteurization at 72 degrees centigrade for 15 seconds and UHT treatment is not sufficient to inactivate the toxin molecule. Staphylococcal enterotoxins along with other toxic shock syndrome toxins are classed as bacterial superantigens. Superantigens bind directly to T cell receptor beta chains without processing. This will be clear from this diagram. Here is a T cell having T cell receptors, and this is an antigen presenting cell having MHC molecule. So, normal antigen binds to MHC molecule and is presented to T cell receptor, okay, TH cell, and this will generate an immune response. This is the reaction which antigen gives, but in case of superantigen, this antigen over here is there, but superantigen will bind MHC and T cell receptor, and this leads to activation of T cells with irrespective of its specification towards a particular antigen. So, when superantigen is present, it will activate a lot of clones of T cell causing reaction, and once bound to MHC class II molecules, SEs stimulate helper T cells to produce cytokines such as the interleukins, gamma interferon and tumor necrosis factor. Among the cytokines, an overabundance of IL-2 is produced and it appears to be responsible for many of the symptoms of staphylococcal gastroenteritis. Staphylococcal enterotoxins are able to penetrate the gut lining and activate local and systemic immune responses. These toxins stimulate the vagus nerve in the abdominal viscera, which transmits the signal to the vomiting center in the brain. The minimum quantity of enterotoxin needed to cause illness in humans is about 20 nanograms. Now coming to symptoms, the symptoms of staphylococcal food poisoning develop within 4 hours of the ingestion of contaminated food, although a range of 1 to 6 hours has been reported, and these symptoms include nausea, vomiting, abdominal cramps, diarrhea, sweating, headache, prostration, and sometimes there is a fall in body temperature that generally lasts from 24 to 48 hours. Treatment for this disease consists of bed rest and maintenance of fluid balance. This disease is self-limiting disease, and just if fluid balance is maintained and if rest is done, this is curable. Now coming to the sources of food contamination, we have discussed that this organism is present on the human body. So food handlers carrying enterotoxin producing Staphylococcus aureus on their noses or on their hands are regarded as the main source of food contamination. They may contaminate food via manual contact or through respiratory secretion. Staphylococcus aureus is also present in food animals and dairy cattle, sheep and goats, particularly if they are affected by subclinical mastitis. Then Staphylococcus will be a contaminant of milk. Air, dust and food contact surfaces can also serve as vehicles in the transfer of Staphylococcus aureus to foods. Staphylococcus aureus may be expected to occur in wide variety of foods which are not given heat treatments. They are prevalent in meats and seafoods. Pork and its products account for many outbreaks. Largest outbreak ever recorded in June-July 2000 in Kansai district of Japan. There were 13,420 victims, and the primary vehicle food was powdered skim milk from a single source. These are the food products involved: milk and its products, chicken a la king, ham both cured and dry, mayonnaise, Minas cheese, water, custard, ice cream pies, cooked beef, rice, rice balls, bean curds. In general, staphylococci may be expected to exist in low numbers in any or all food products that are from animal origin or that are being handled directly by humans. Coming to ecology, staphylococci do not compete well with the normal flora of most of the foods, and this is especially when large number of lactic acid bacteria are present in the food product. So competition is there, and Staphylococcus is unable to compete with those lactic acid bacteria. A large number of investigators have shown the inability of Staphylococcus aureus to compete in both fresh and frozen foods at temperatures that favor staphylococcal growth. The normal food saprophytic biota offers protection against staphylococcal growth. So, जो normal saprophytic food biota is present, it grows at a higher rate and it can inhibit Staphylococcus aureus present in food. Now coming to prevention, when susceptible foods are produced with low numbers of staphylococci, they will remain free of enterotoxins and other food poisoning hazards if these are kept either below 4.4 degrees centigrade or above 60 degrees centigrade. So, basically the growth range of Staphylococcus is to be avoided. The temperature at which bacteria can grow should be avoided for storage of food, and susceptible foods should not be held within this growth range for more than 3 to 4 hours. Now, another prevention is to have good sanitation, then using of ingredients free of staphylococci, such as use of pasteurized milk rather than raw milk, and not allowing employees with staphylococcal infections to handle food products. Killing of staphylococci in foods can be one of the measure, and prevention of the growth of staphylococci can be carried out, and this is usually refrigeration. So, principally the growth temperature of the bacteria should be avoided, so that it is not able to proliferate in the food product and produce toxin. Now, these are the references which have been used for preparation of this presentation. In this paper, Argudin et al. On food poisoning and Staphylococcus aureus enterotoxins give a good view of staphylococcal enterotoxins and staphylococcal enterotoxin like antigens. Thank you. I am Dr. Pavas Goswami from Bhaskaracharya College of Applied Sciences, and today I will be delivering a lecture on food intoxication, and in this I will be covering Clostridium botulinum. So, contents of my lecture will include history, then Clostridium botulinum, its characteristics, medium for isolation, serological types, distribution of this bacteria, growth and toxin production, botulinum neurotoxin, foods involved, adult botulism syndrome, botulism outbreak, and infant botulism. So, coming to history, an outbreak of sausage poisoning occurred in 1793 in Wildbad, Württemberg, Germany, where 13 cases of food poisoning were there, and out of these 6 deaths took place. It was traced to blood sausage, which are pig gut filled with blood and other ingredients. The filled gut was tied, boiled, briefly smoked and stored at room temperature. Between 1820 and 1822, Justinius Kerner studied 230 cases of sausage poisoning in Württemberg and noted that the product did not become toxic if air pockets were left in the casing. In 1896, 24 music club members in Elizalde ate raw salted ham, out of which 23 became ill and 3 died. When Armengen of the University of Ghent studied the outbreak, he found that the ham was neither cooked nor smoked, and the same organism was recovered from the ham and the spleen of victim, suggesting that this organism was responsible for the food poisoning and also for the death of the victim. When Armengen named the causative organism as Bacillus botulinus, and in this botulinus comes from sausage, Latin is Clostridium botulinum. This is gram positive rod, saprophytic, forms spores, gas forming, and an anaerobic organism. These are gram positive, and this suggests again high concentration of peptidoglycan in their cell wall and low concentration of lipids. Medium for isolation of Clostridium botulinum have casein enzymic hydrolysate, yeast extract, dextrose, disodium phosphate, sodium chloride, magnesium sulfate, agar, and pH is around 7.4 plus minus 0.2. To this basal medium, egg yolk emulsion is added. This basically gives an idea of lecithin production by Clostridium botulinum, and antibiotics such as cycloserine, sulfamethoxazole and trimethoprim are also added to make the medium selective for Clostridium botulinum, so that other microorganisms or other bacteria cannot grow. So, these antibiotics are added, to which Clostridium botulinum is resistant and its growth is not affected by these antibiotics. 7 types of Clostridium botulinum have been distinguished on the basis of serological specificity of their toxins. Type A cause human botulism in western part of United States. This is more toxic than type B. Type B is found more often than type A in most soils of the world. This is less toxic to humans. Type C causes botulism of fowls, cattle, mink and other animals. Type D is involved in forage poisoning of cattle, especially in South Africa. Type E is toxic to humans and obtained chiefly from fish and fish products. Type F cause human botulism and have been isolated from Denmark. Type G is not implicated in human botulism and have been isolated from Argentina. Clostridium botulinum strains are frequently divided into 3 general groups. Group 1 includes all type A strains which are proteolytic, and proteolytic strains of types B and F. Group 2 includes all type E strains including all non-proteolytic and non-proteolytic strains of B and F. Group 3 includes type C and D. They are non-proteolytic and share a common metabolic pattern. Distribution of Clostridium botulinum. These spores are occurring in soil and they are found worldwide. The non-proteolytic types are associated more with waters than soil. They are found in lakes and other water bodies. As compared to soils, they are present more in water. Proteolytic strains of Clostridium botulinum are known to have several characteristics, such as they digest casein, produce hydrogen sulfide. Temperature range for their growth is 10 to 50 degrees centigrade, and these are more heat resistant. Hydrogen sulfide production leads to putrefaction of the food product, and food which has been contaminated with Clostridium botulinum can easily be detected, because hydrogen sulfide production will be there. However, non-proteolytic strains may not produce gas. Non-proteolytic strains of Clostridium botulinum ferment mannose. They are favored in their growths by carbohydrates, and the temperature range for their growth is 3.3 to 45 degrees centigrade, and these strains are less heat resistant as compared to other strains. Growth and toxin production. Toxin production depends on the ability of cells to grow in food and autolyze there. A, B, E and F toxins are synthesized as large comparatively inactive proteins which become fully toxic after some hydrolysis. So, when toxin is produced, it may not be toxigenic and it may require some further treatment with proteinases or other enzymes to increase its toxicity. Factors influencing spore germination, growth and toxin production are composition of food, moisture content, pH, oxidation reduction potential, salt content, temperature and time of storage of food. So, I have discussed that there are several factors which affect bacterial growth, and these factors include growth temperature, then optimum temperature, pH, optimum pH, pH range, then oxidation reduction potential. So, several factors are there which can affect what kind of carbohydrates or proteins are present in food, whether bacteria will able to utilize them or not. So, all these factors are present affect the bacterial growth, and in this an important factor is composition of food. Glucose or maltose are considered essential for toxin production by Clostridium botulinum. Milk or casein is also responsible for toxin production, and toxin production have also been reported for corn steep liquor. Potency of toxin in canned foods in descending order is as follows: corn, peas, string beans and spinach. Moisture content is another important factor which affects the growth and toxin production. Experiments on dehydrated meats have shown that toxin production occurs rapidly at 60% moisture, then at 40%, and it was found that toxin production was inhibited at 30% moisture. So, at 30% moisture or below, toxin production was not reported. Highest toxin production was reported at 60%, while a lower amount of toxin was produced at 40% moisture. Salt content of food also affect the toxin production. Under favorable conditions for growth, 8% or more salt is required to inhibit Clostridium botulinum. Another important factor is pH. Neutral pH favors the growth of Clostridium botulinum. pH of 4.5 or lower will prevent toxin production in most of the foods. Under very acidic conditions, this organism is unable to produce toxin. pH for spore germination is higher, that is 5 in bread, and maximal pH of 8.89 was found suitable for vegetative growth. Though pH of 4.5 or less prevent toxin production, still Clostridium botulinum toxin have been found in inadequately heat processed high acid foods having pH less than 4.5. These include tomatoes, tomato juice, blackberries. Possible explanation of this fact are perhaps there are some other microorganisms growing in that food and they are increasing the pH and decreasing the acid content. Thus, they are making conditions suitable for the growth of Clostridium botulinum. Similarly, if the pH of food is highly alkaline, then growth of microorganisms may decrease this pH and may bring it within the range of the growth for Clostridium botulinum and toxin production. Variation and stratification of the pH may also occur in acidulated product to permit the growth of Clostridium botulinum. Now, temperature. Vegetative growth takes place at a lower temperature than the minimum for spore germination. Usually, lowest temperature of growth is around 10 to 11 degrees centigrade, while maximal temperature for growth is 48 degrees centigrade. Type E organisms may produce gas and toxin within 31 to 45 days at temperature as low as 3.3 degrees centigrade. This is again an important fact. Now you can observe that at temperature of 3.3 degrees centigrade, the bacteria still it is producing toxin, though time for toxin production is more from 31 to 45 days, but still it is not inactive and toxin production have been reported at such low temperatures. Optimal temperature for toxin production and growth differs for both proteolytic and non-proteolytic strains. It is 35 degrees centigrade for proteolytic strains and 26 to 28 degrees centigrade for non-proteolytic strains. Now coming to an important part of this talk, Clostridium botulinum toxin. Clostridium botulinum toxin is a very potent neurotoxin. This is formed within the organism and it is released on autolysis. The genes for botulinum neurotoxin are present on chromosomal DNA in case of A, B, E and F, and type G botulinum neurotoxin is plasmid borne. Toxin is absorbed into the bloodstream through the respiratory mucous membranes as well as through the walls of the stomach and intestines. Botulinal toxin is heat sensitive and may be destroyed by heating at 80 degrees centigrade for 10 minutes or boiling temperatures for a few minutes. Botulinum neurotoxin is produced as a single polypeptide chain. Post translationally, it is nicked to form two chains. One is the heavy chain and other is the light chain. Heavy chain is of 100 kilodaltons and light chain is of 50 kilodaltons. The two chains are held together by disulfide bond. Toxin inhibits the release of acetylcholine neurotransmitter from nerve cells. This will be clear from this diagram. Say this is the nerve terminus and muscle cell. This depicts the nerve cell and the muscle cell. Between these two cells there is synaptic cleft. There are acetylcholine receptors present on the muscle cell. These acetylcholine receptors bind to the acetylcholine molecules which is released from the nerve cells. Okay. So, under normal conditions what happens is synaptic fusion complex is formed. The main role of this complex is to fuse this vesicle which is containing acetylcholine, which is a neurotransmitter, with nerve terminus. So, when this synaptic fusion complex is formed, this vesicle fuses and acetylcholine from the nerve is released, which is sensed by the receptor, and in this way signal is transmitted. This is the normal condition, but in presence of Clostridium botulinum toxin the scenario changes. Clostridium botulinum toxin is endocytosed by the nerve cell, and as we have discussed, it is having two chains. One is a heavy chain and another is a light chain. Heavy chain is involved in endocytosis of the toxin into the cell, nerve cell, and once this toxin is endocytosed, the light chain from this vesicle is released and it kind of degrades the synaptic terminus fusion, and this degradation inhibits the fusion of vesicle which is carrying acetylcholine molecules with the nerve terminus, and this fusion is inhibited due to degradation of synaptic complex by light chain of the botulinum toxin. And once this happens, this nerve transduction do not take place and this process is inhibited. So, this is the mode of action of Clostridium botulinum toxin. Components of synaptic fusion complex are there. There are several proteins. These include synaptobrevin. Types B, D, F and G act at synaptobrevin. SNAP25 is another protein which is inactivated by types A, C and E, and syntaxin of the synaptic fusion complex is also inhibited by type C. Foods involved in this Clostridium botulinum toxin production are usually these are the inadequately processed home canned foods. These include string beans, sweet corn, beets, asparagus, spinach, tomatoes, chili peppers, mushroom, sausages and fish. All those foods which have not been properly heat treated may produce and are infected and are contaminated with Clostridium botulinum may produce toxin. So, growth of Clostridium botulinum in some foods results in foul rancid odor which leads to their rejection. So, one can detect if foul and rancid odor is coming from the food, there are chances that Clostridium botulinum is present and toxin is occurring in the food. Meats and proteinaceous low acid vegetables develop an obnoxious odor and again indicates presence of Clostridium botulinum. More acid foods and those in low proteins may become toxic without the evidence of proteolysis. Now, this is dangerous because food is not spoiling as such. So, we cannot know that whether toxin is there or not. Non-proteolytic strains also give less evidence of spoilage, and these strains again, if evidence of spoilage is not there, there are chances that the food contaminated with toxin may be taken up or may be utilized by people. Gas production is also not always evident and hence it is not a reliable indication of spoilage by this organism. Coming to the syndromes caused by botulinum, there are two syndromes. One is adult botulism syndrome and another is infant syndrome. So, adult botulism syndrome is due to ingestion of food having Clostridium botulinum toxin, and their symptoms develop between 12 to 72 hours of ingestion of contaminated foods. Symptoms of adult botulinum syndrome are acute digestive disturbance, nausea, vomiting, fatigue, diarrhea, dizziness, headache, dryness of skin, mouth and throat, and then constipation may also take place. Initially diarrhea may be there, but later on it may be followed by constipation. There is paralysis of muscles, double vision, difficulty in swallowing and speaking, tongue of the patient may become swollen and coated, respiratory failure is there followed by death. Treatment of adult botulinum syndrome is administration of botulinum antitoxin at the earliest. If this administration is delayed, then botulinum antitoxin may not work. Then artificial respiration can be carried out, and maintaining fluid balance in the body is also a part of this treatment. Coming to conditions necessary for an outbreak, first the food may be contaminated and this food product should allow the growth of Clostridia and produce toxin. Inadequate heat treatment during canning may lead to growth of Clostridium botulinum. Environmental conditions, if not controlled, and these conditions after processing, if they permit the spore germination and growth of toxin production, growth and toxin production by organisms, then outbreak can occur. Insufficient cooking of the food to inactivate the toxin is also one of the reason for outbreak. Maintaining good sanitation throughout the production and handling can be carried out for prevention of outbreak. Use of approved heat processes for canned foods, rejection of all gassy or spoiled canned foods, refusal even to taste a doubtful food. If there is a doubt that toxin may be present, the food should not be tasted because the toxin is active at nanogram levels of quantity. Then avoidance of foods that have been cooked, held and not well reheated. If food is held at ambient temperature, then growth of Clostridium may occur and toxin production may also occur, and if food is not reheated again, then toxin inactivation may not take place. Now, another syndrome is infant botulism. This disease is different from foodborne botulism. In this disease, toxin is released in vivo. Toxin is not coming through food, but toxin is being formed in the intestine, and symptoms include constipation, weakness, lack of sucking by the infant and diminished gag reflux. Vehicle food for Clostridium botulinum is honey. Spores of Clostridium botulinum are present in honey. Actually, what is happening in infants, intestinal microflora is not able to inhibit the growth of botulinum, Clostridium botulinum, and this results in toxin production. However, in adults, the intestinal microflora is well established and Clostridium botulinum is not able to compete with this intestinal flora, and so this disease does not take place in adults. In adults the disease is due to foodborne intoxication, while in infants Clostridium botulinum may multiply as the microflora is not stabilized. It does not provide resistance to the growth of this bacteria, and this bacteria will multiply in the intestine, produce toxin and cause infant botulism. These are the references which have been used for this preparation of this lecture and presentation. Thank you.
On that note, I would like to thank sir for this very enriching discussion, and I hope, dear friends, that you have come to understand what various kind of food intoxications are and what are their cures and what needs to be done instead of for avoiding such food intoxications. On that note, dear friends, thank you for watching our show. Stay tuned and keep watching. Thank you.